Good evening. I am Madhurima Datta, a Zoology and Biology educational content creators. Today's content for B.sc, M.Sc and also CSIRNET aspirant students. Today's paper is non-coded and diversity of life form, and topic is the phylogenetic reconstruction. So, taxonomic characters and phylogenetic reconstruction. What is phylogeny? A major goal of systematics is to infer the evolutionary tree or phylogeny that relates all extant and extinct species. This task is accomplished by identifying organismal features formally called characters that vary among species. Phylogenetic analysis depends upon finding among organisms shared features that are inherited from a common ancestor. Next, homology. Which is homology? Homo means same. So, character similarity that results from common ancestry is called homology. Similarity does not always reflect common ancestry. Independent evolutionary origin of similar features on different lineages produces patterns of similarity among organisms that do not reflect common descent. Next, homoplasy. Character similarity that misrepresent common descent is called non-homology or homoplasy. Endothermy of birds and mammals is an example of homoplasy. This condition separates them the lineages of birds and animals. Next point is using character variation to reconstruct phylogeny. At first, to infer the phylogeny of a group using characters that vary among its members, the first step is to determine which variant form of each character was present in the common ancestor of the entire group. Next, this character state is called ancestral for the group as a whole. We presume that all other variant forms of the character arose later within the group, and these are called evolutionary derived character states. Next, determining the polarity of a character refers to identifying which one of its constructing states is ancestral and which ones derived. Next, the method used to examine the polarity of variable character is called outgroup comparison. We consult an additional group of organisms called an outgroup that is phylogenetically close but not within the group being studied. Example, amphibians and differing groups of bony fishes constitute appropriate outgroups to the amniotes for polarizing variation in the dentition of amniotes. Next, teeth are usually present in the amphibians and bony fishes. Therefore, we infer that presence of teeth is ancestral for amniotes and absence of teeth is derived. The polarity of this character indicates that teeth were lost in the ancestral lineage of all modern birds. Next, clades. Organisms or species that share derived character states form subsets within the study group called clades. Greek klados means branch. Next, synapomorphy. A derived character shared by the members of a clade is formally called a synapomorphy. Greek means synapses joining together and morph means a form of that clade. Example, among the extant amniotes, absence of teeth and presence of feathers are synapomorphies that identify the birds as a clade. Next, nested hierarchy. The pattern formed by the derived states of all characters within our study group takes the form of a nested hierarchy of clades within the clades. Next, plesiomorphic and symplecomorphic. Character states ancestral for a taxon are often called plesiomorphic for the taxon, and the sharing of ancestral states among organisms is termed symplecomorphy. Next, cladogram. The nested hierarchy of clades is presented as a branching diagram called a cladogram. Next, comparative cytology. Comparative cytology uses variation in the number, shapes, and size of chromosomes and their parts To obtain the variable characters for constructing cladograms, comparative cytology is used almost exclusively on living rather than fossilized organisms. Next, phylogenetic tree. Taxonomists often make a technical distinction between a cladogram and a phylogenetic tree. The branches of a cladogram are only a formal device for indicating the nested hierarchy of clades within the clades. The cladogram is not strictly equivalent to a phylogenetic tree, whose branches represent real lineages that occurred in the evolutionary past. In here, according to jaw, there are two types, agnatha and gnathostomata. Agnatha, they are jawless like hagfish, and gnathostomata, they possesses jaw. They are all are the gnathostomata, they possesses jaw. Then, according to lungs, in salamander, they have proper lung system, but in birds have no lung system. Then, according to claws and nails, they are branches are increases like lizards, pigeon, mouse, and sheep. They consume claws and nails. Feathers. Pigeon possesses feather, and next fur. Mammals and mammary glands. They are mouse and chimpanzee have mammary glands, but pigeon possesses feather but do not possesses mammary gland and fur. Today's topic has ended.